Hello everyone. In continuation to our topic research methods, the next is Latin square design, shortly called as LSD. A Latin square design is an experimental design used to evaluate the effects of multiple treatments, that is factors, on a response variable. It is a balanced design that controls for two extraneous variables, ensuring each treatment is tested under the same conditions. Example, consider an experiment testing four different fertilizers A, B, C, D on crop yield across four different fields and during four different time periods. The Latin square design ensures that each fertilizer is tested in every field and during every time period exactly once. The arrangement might look like this. So here we may notice time 1 to time 4, field 1 to field 4. In this field 1, you may notice A, B, C, D, all the treatments are occurring, but at different time intervals. Same like all the fertilizers are occurring at different time intervals, even in the field 2. Like that, it is arranged. Features of LSD: control of two variables. Each treatment appears once in each row and each column, controlling for two sources of variability, that is, time and location. Balanced design. All treatments are tested equally across all conditions. Symmetrical arrangement ensures that the experimental conditions are symmetrically arranged. Requires a square number of treatments like 4, 9, 16, like that. Advantages of LSD: it reduces variability, controls two sources of variation, increasing the precision of the experiment. Second advantage is efficient use of resources, minimizes the number of experimental units needed. Third one, balanced comparison. Each treatment is equally exposed to each condition, reducing bias. Suitable for complex experiments, ideal for experiments where two factors need to be controlled simultaneously. Disadvantages of LSD are complexity. Design and analysis can be complex and may require advanced statistical techniques. There are strict assumptions, assumes that row and column effects are additive and do not interact, which might not be always true. Limited applicability, only practical when the number of treatments is equal to the number of levels of the control variables. Missing data values, if the data is missing for any treatment in a row or column, it can complicate the analysis. Next design is factorial design. The factorial design is an experimental design that involves testing the effects of two or more factors, that is independent variables, on a response variable, which is the dependent variable. It examines the individual and combined effects of the factors. Let us consider this example 1. Suppose we want to study the effect of temperature, that is factor A 20 degrees, 30 degrees, and 40 degrees Celsius, and the pH factor B 5, 7, and 9 on the growth of a specific plant. A 3 by 3 factorial design would involve 9 combinations, 20 degrees, 30 degrees, 40 degrees. Here in columns pH 5, 7, and 9. So here you will have this A1, B1, A1, B2, A1, B3, A2, B1, A2, B2, A2, B3, A3, B1, A3, B2, and A3, B3. Another example, let us design a 3-factor experiment to investigate the effects of 3 different factors on the yield of a particular crop. The factors and their levels are: first factor is factor A, type of fertilizer. Is it organic or inorganic? We are calling it as A1 and A2. Amount of water, factor B. Low, medium, high, which is B1, B2, B3. Sunlight exposure, factor C. C1 and C2, 4 hours per day, 8 hours per day. In a full factorial design, all the possible combinations of these levels will be tested. This results in 2 into 3 into 2, we get 12 treatment combinations. So the set of for the experiment will be to ensure reliability and account for variability. Multiple replicates should be used for each treatment combinations, means the repetitions. For simplicity, let us assume 3 replicates per treatment group. So the treatment combinations and replicates will be, we have A1, B1, C1, A1, B2, B1, C2, A1, B2, C1, A1, B2, C2, A1, B3, C1, A1, B3, C2, A2, B1, C1, A2, B1, C2, A2, B2, C1, A2, B2, C2, A2, B3, C1, A2, B3, C2, like that. So what is A1? That is our organic. So organic will come. B1 is low and C1 is 4 hours per day. So we are writing that organic low 4 hours per day. You are going to do 3 replicates of it. Likewise, against this each of the treatment group, you are expanding what exactly it is and writing it. So this is our treatment combinations and its replicates. So after you decide about this, measure the crop yield, that is weight of the produce for each replicate in each treatment group after a certain growth period. Analyze the data to determine the main effect of each factor A, B, and C, the two-way interaction effects between factors A, B, A, C, B, C, the three-way interaction effect, that is A, B, C. This can be done using analysis of variance to assess the significance of the effects and interactions. The features of this particular design, factorial design, is multiple factors test the effects of 2 or more factors. Factor combinations, that is, examines the individual and combined effects of factors. Balanced design, each factor is tested at every level of other factors. Interaction effects reveals how factors interact with each other. Advantages of this design: it is efficient because it tests multiple factors simultaneously. It is comprehensive because it examines individual and combined effects. It reveals interactions because it helps us to understand how factors interact. It is generalizable because results can be applied to various situations. Disadvantages of this factorial design: it is complex, large number of factor combinations, resource intensive, it requires more resources, time, money, and materials. Analysis is difficult because it requires specialized statistical analysis, assumes no confounding variables, assumes no extraneous variables, affect the results. So these are the disadvantages.